Hello everybody and welcome to our module on cardiac muscle. Cardiac muscle shares many similarities with skeletal muscle. Both cardiac and skeletal muscle are striated. They both contain sarcomeres and actin and myosin and troponin and tropomyosin, and they both have T-tubules that abut the sarcoplasmic reticulum. And I talk about all these things in detail in the video on skeletal muscle. What I'm going to do in this video is focus on the differences between cardiac muscle and especially what makes cardiac muscle unique. One way that cardiac muscle differs from skeletal muscle is that cardiac muscle has dyads instead of triads. In the video on skeletal muscle, I talked about the triad shown on the screen here. A triad consists of a T-tubule down the middle surrounded by two terminal cisternae of the sarcoplasmic reticulum. This entire structure is called a triad. In cardiac myocytes, you have a T-tubule, but it's only surrounded by a terminal cisterna on one side, not the other, and therefore this is called a dyad instead of a triad. Some other differences are shown on this slide here. Cardiac muscle is involuntary. You can't make your heart beat the way you can with skeletal muscle. In addition, skeletal muscle is depolarized by motor neurons. Cardiac muscle is depolarized by pacemaker cells like the SA node. And then finally, cardiac muscle has gap junctions, which are shown in this drawing on the screen here. Gap junctions are connections between two neighboring cells so that molecules can move from one cell to the other without having to be excreted into the extracellular space. And this is very important because this is what allows depolarization to spread from one cell to another, so that once one part of the heart begins to depolarize, a wave of depolarization spreads over the entire heart. This is not true of skeletal muscle tissue. Each skeletal muscle fiber has its own motor neuron that has to tell it to depolarize. Cardiac muscle also has a different action potential. In the video on skeletal muscle, I talk about the skeletal muscle action potential, which looks like this shape on the bottom left side of the screen here. In cardiac muscle, there is a phase 2 not present in skeletal muscle. Phase 2 is caused by calcium influx into cardiac muscle cells via L-type calcium channels. This is phase 2 shown here. It's this plateau portion of the action potential that is unique to cardiac muscle, caused by calcium influx through calcium channels. Let's talk about cardiac L-type calcium channels in a little bit more detail. These calcium channels are also called dihydropyridine receptors, and they get that name because they can bind dihydropyridine calcium channel blocking drugs like amlodipine and nifedipine. And you may know that these drugs are sometimes used in the treatment of hypertension. But here's the confusing part that you have to understand. Even though the L-type calcium channels in the heart are called dihydropyridine receptors, and even though they can bind these drugs, they do so with a very low affinity, and as a result, amlodipine and nifedipine and other dihydropyridine calcium channel blockers have very little effect in the heart. Most of their effect is in smooth muscle. In contrast, non-dihydropyridine calcium channel blockers like diltiazem and verapamil have a much higher affinity for the L-type calcium channels in the heart, and therefore these are the drugs that are used when you want to block calcium channels in the heart. So you've got to imagine that you have a cardiac myocyte and it's got an L-type calcium channel on its surface that will allow calcium to enter the cell. This influx of calcium through the L-type calcium channels is very important for contraction of cardiac myocytes, and this is a distinction from skeletal muscle. In skeletal muscle, all the calcium comes from the sarcoplasmic reticulum. Calcium influx through channels on the plasma membrane is not important. It's different in the heart. This calcium influx through these channels on the surface of the cell triggers the sarcoplasmic reticulum to release calcium via the ryanodine receptor. This is called calcium-triggered calcium release. It's unique to cardiac myocytes, and because of this phenomena, this phase 2 of the action potential is very important. Calcium comes into the cell in this phase of the action potential that triggers the sarcoplasmic reticulum to release calcium, and that's what makes the myocyte contract. Very different from skeletal muscle, where you have a motor neuron depolarizing the muscle cell, and that causes the sarcoplasmic reticulum to release calcium that leads to contraction. Let's talk about contractility. With each heartbeat, all cardiac muscle cells contract. So if the heart wants to squeeze harder and raise the contractility, it can't recruit extra fibers. That's what skeletal muscle does sometimes, but the heart can't do this. The only way the heart can squeeze harder is by sending more calcium into the cell. This will lead to more contraction. The way this is done is the heart allows more calcium entry via L-type calcium channels. That leads to more calcium-triggered calcium release from the sarcoplasmic reticulum, and this will lead to more contraction and higher contractility. In your textbooks, this is referred to as an increased calcium transient. This is just a fancy way of talking about more calcium in the cell. It's called the calcium transient because when you measure calcium concentration in a muscle cell, you see a transient rise like this, and when there's more contractility, the rise gets bigger. That's called an increase in calcium transient, and that's what leads to more contraction. So if you understand that, then it's very easy to understand what the non-dihydropyridine calcium channel blockers do to cardiac myocytes. These are drugs like diltiazem and verapamil, and as I told you before, these are the drugs that block the L-type calcium channels in cardiac myocytes. They will decrease contractility because they block calcium entry into the cell. Therefore, they are negative inotropes on the heart. They also have properties that allow them to slow conduction and lower heart rate. This is because of effects they have on cardiac pacemaker cells. But the point here is that by blocking these L-type calcium channels, you decrease contractility because there's less calcium-triggered calcium release from the sarcoplasmic reticulum. So blocking calcium channels can decrease contractility, and in contrast, activating the sympathetic nervous system can increase contractility. So let's talk about how this happens. The sympathetic nervous system works through G proteins that are coupled to sympathetic nervous system receptors. These G proteins raise the intracellular cyclic AMP levels by activating the enzyme adenyl cyclase. If you look at the bottom of the screen, adenyl cyclase converts ATP into cyclic AMP. Cyclic AMP in turn increases the activity of the enzyme protein kinase A, and protein kinase A phosphorylates calcium channels, and this leads to more calcium in the cell. And as we've been discussing, more calcium into cardiac myocytes leads to more contraction. This is how the sympathetic nervous system raises contractility. And here's a picture just to make this more clear. So cardiac muscle cells have GS proteins on their surface. The S stands for stimulatory, and that's because they stimulate the enzyme adenyl cyclase. Adenyl cyclase raises the intracellular level of cyclic AMP. That activates protein kinase A, which phosphorylates calcium channels. That allows more calcium into the cell, and that leads to more calcium-triggered calcium release, and that leads to more contraction. And this is how beta-1 receptors work. You may know that stimulation of beta-1 receptors in the heart leads to increased contractility, and this is the way it happens. The beta-1 receptors are linked to GS proteins that raises cyclic AMP levels. That leads to more calcium and more contractility. Lusitropy is the process of myocardial relaxation. This is the opposite of contractility, and it's an active process. The heart doesn't relax just by ceasing contraction. It actually undergoes an active process called lusitropy so that it can quickly relax and be ready for the next heartbeat. And increases in lusitropy always accompany increases in contractility so that when the heart beats faster, it also relaxes faster. Lusitropy is mediated by a structure called the SERCA. It's found in the membrane of the sarcoplasmic reticulum, and it transfers calcium from the cytosol back into the sarcoplasmic reticulum. This is an active process. It's an ATPase that uses ATP hydrolysis to quickly bring calcium back into the sarcoplasmic reticulum and get ready for another contraction. And because of the SERCA and the process of lusitropy, heart myocytes can rapidly contract and relax. The SERCA is regulated by a protein called phospholamban, and the sympathetic nervous system uses this regulatory protein to modify lusitropy just like it modifies contractility. Phospholamban is an inhibitor of the SERCA, and it's phosphorylated by beta-adrenergic stimulation via protein kinase A. And when it's phosphorylated, it stops inhibiting SERCA, and the SERCA can work faster to take up calcium and cause relaxation. And here's a picture just to make this more clear. Sympathetic stimulation of cardiac myocytes leads to phosphorylation of phospholamban, as shown in this picture here on the screen. This inactivates phospholamban and relieves the inhibitory effect. This allows SERCA to uptake more calcium and work faster. So the bottom line here is that the sympathetic nervous system can increase both contractility and relaxation in the heart, both by modifying the amount of calcium in the cytosol. That's the big picture take-home point. And that concludes our video on cardiac muscle.